Hi, everyone. Thanks for coming to our agroecology intern double feature today. Our first speaker is Flynn Hibbs. Flynn graduated from Hampshire College in Massachusetts with a Bachelor of Arts with a concentration in wildlife conservation and animal behavior. Her past research has focused on invertebrates, and she looked at the effects of solar arrays and also did an intensive survey in the Chihuahuan desert. After her internship, Flynn's going to work at the Montana Conservation Corps, and we have really enjoyed having Flynn in the lab for the past 10 months. She's really hardworking, easy to get along with, friendly, and just fun to be around. Today, she will give a talk entitled Effects of Soil Moisture and Sunlight on Plant-Plant Interactions in Semi-Native Subtropical Rangelands. Flynn, over to you.
Awesome. Thank you so much for that sweet introduction, Betsy. As soon as my slide changes, I'll get into it. Okay. So hopping in, my role on the ranch while an intern here was to help launch a precipitation manipulation experiment by helping with the construction of rainout shelters and irrigation systems. And rainout shelters, which are pictured here, are these built structures that help us mimic the effects of drought by limiting the rainfall that reaches the ground. And we built a system that then diverts the water collected off of the roof to a storage tank that then gets irrigated to other plots that are without these roofs. And this is to mimic extreme rainfall events. So the goal of this experiment is to understand how various predicted climate scenarios are going to impact and change ecosystem function of rangelands in South Central Florida. And the hope is that these results can help inform management plans for ranchers that are going to need to adapt their operations to the realities of climate change. So for my intern project, I wanted to conduct research within the scope of the rainout experiment and specifically focus on plant interactions and plant growth and how they respond or change when subject to precipitation treatments in this experiment. So a little background on plant interactions. They range from competition to facilitation. So a facilitative interaction means plants are providing benefits to one another. And it can be through many different mechanisms like herbivory or protection from herbivory, and shading. And according to the facilitation theory, the more abiotic stress plants are subject to, the more common these facilitative interactions are going to be. So we may see more facilitative interactions after extreme weather events. However, some studies have seen the opposite and actually saw competitive interactions increase with extreme weather conditions. So we know that plant interactions are going to play a really important role in how ecosystems respond to climate change. And these interactions could maybe be able to mitigate the effects of climate change or extreme weather, but it's just not well understood yet. And this rainout project or experiment really provides a great platform to begin to uncover what these interactions are going to look like through the simulated climate scenarios that amplify extreme weather events. So to measure plant responses in response to changes in their environment, we can look at a bunch of different things. One thing we can look at is survival over a study period. We can also look at plant growth, so things like change in height or number of leaves, number of flowers or inflorescence or tiller count, etc. We can also do it by examining leaf traits like specific leaf area and leaf dry matter content. And so these two things influence a plant's relative growth rate. So specific leaf area, or SLA, determines a plant's net photosynthetic rate and relative growth rate, whereas leaf dry matter content is going to indicate leaf tissue density and the capacity to retain nutrients or resources. And plants can modify their leaf traits in response to changes in their environment. So I had research questions about these plant responses and the plant interactions in these two areas. One about the abiotic conditions and how it would affect them. So things like soil moisture and sunlight, and also how competition would affect these plant responses and plant interactions. So moving into my questions and hypotheses. One question we had was how these abiotic conditions were going to affect plant survival and growth. So in terms of soil moisture and how it would affect it, we expect to see a non-linear relationship between plant growth and soil moisture. So what I mean by that is, as we see soil moisture increase, we're going to see plant growth increase, but then we're going to reach a certain point where there's too much soil moisture and it's detrimental to the plants and their growth. And we're going to see plant growth decline, or we're going to see plant death. In terms of how sunlight would affect this, we expect to see the plants receiving more sunlight to have greater survivability and greater growth. And then in terms of how soil moisture and sunlight would influence our plant traits, we expect to see increased LDMC and reduced SLA when soil moisture is high. So when there's a waterlogging of the soil, it can result in physiological drought for plants because it creates an oxygen deficiency in the soil, which will cause the plants to increase their LDMC and reduce their SLA to prevent water loss. And we expect to see the opposite when there's low soil moisture. So a decrease in LDMC and an increase in SLA. In terms of sunlight, we expect the plants to respond to high levels of sunlight by increasing their LDMC and reducing their SLA, and vice versa when there's less available sunlight. So if there's lots of available sunlight, plants likely don't need to maximize their photosynthetic rates by adapting and increasing their leaf area, whereas the plants receiving less light would want to increase their leaf area to counteract that limiting factor to their growth. And then in terms of how the abiotic conditions would influence our plant interactions, we expect to see facilitative interactions in stressful pastures and competitive interactions in the least stressful pastures. So by stressful, I mean their environmental conditions. So if there's dries or more wet beds, things like that. And then in terms of how competition influences plant growth, we expect to see greater plant growth among our individuals without competitors compared to those that have competitors surrounding them. We believe this will happen because individuals without competitors are going to have more access to nutrients in the soil, more available sunlight. That'll allow for greater growth. And then in terms of how competition would influence plant leaf traits, we expect to see lower LDMC and higher SLA in plants without neighbors, as they're going to have more access to resources and they won't need to retain their nutrients and can instead focus on increasing their leaf area and therefore their photosynthetic rates. We expect the opposite in plants with the neighbors, so high LDMC, low SLA. And then how would competition influence our plant interactions? So again, as before, we expect to see our plant interactions mainly influenced by the amount of stress these plants are under in their given plot or pasture. So now moving into my methods. So originally I was going to implement the rainout treatments into my project as we had planned to have the rainout experiment up and running this summer, but it turns out that building maybe one of the largest rainout experiments ever attempted takes a really long time. And we thought it was going to be ready and it wasn't. So a couple months into my project, I dropped the precipitation treatments from my experimental design after starting my project. So even though my project didn't have these precipitation treatments, I was still able to get preliminary data on how abiotic conditions and competition influenced these plant interactions and plant responses, which could hopefully inform and provide context for future results from the rainout experiment. So what you're looking at on this slide is the experimental design of a rainout pasture, and the plots circled are where I chose to conduct my research with the precipitation treatments in mind. And hopefully this will provide context as I continue talking about my methods. So zooming out a little bit, there are 8 experimental pastures in the rainout experiment. 4 pastures are improved, meaning they have low species diversity and are planted with these non-native grasses with high forage quality. And 4 of the pastures are semi-native, what we call semi-native, meaning they have high species diversity and they're not ditched as heavily and get flooded for longer. So I chose to conduct my research in these semi-native pastures. And the pastures here are along an elevation gradient, which are labeled, they're labeled to reflect this. So the lowest elevation was found in pasture 1, the highest elevation is at pasture 4. So we've got 4 pastures. We have 3 plots in each pasture and 4 individuals in each plot. So that's 48 individuals total that I was following throughout my study. And so if we zoom in on one plot, what we have here is the 4 individuals in a plot. And when I went out to these plots, I made sure to select pairs of similar sizes and leaf counts. So we've got 2 species and 1 in the pair has no neighbors. So I would find my focal individual and I would clip 20 centimeters around it so it was not surrounded by any other plants. hence being neighborless. And I would do that clipping each month when I visit it. So that looks like this. On the left, we've got the plant with neighbors, and on the right, we've got a plant without. In terms of the species I looked at, I chose 2 common palatable grass species that are found in semi-native grasslands. And they had to be common enough that I could find them in all of the 5-meter by 5-meter plots. So one species I focused on was Axonopus vircatus, or big carpet grass. It looks like this. And I also focused on Dicanthelium weberianum. It's super cute inflorescence. So in terms of my field measurements, each month I recorded plant height, number of leaves, and soil moisture at each plant. And also I collected data monthly from August to November. I forgot to mention that. I also recorded photosynthetically active radiation, aka PAR. Also, we can think of it as the available sunlight that the plants can use for their photosynthesis. And that was recorded opportunistically, as it's really hard to get this measurement in the wet season when there's often clouds covering the sun. So I also recorded leaf traits. So at the end of my study, I collected leaves from all of my surviving plants. I scanned them, measured their fresh weight, dried them, got their dry weight, and then used a program called ImageJ to calculate their leaf area. So for to understand or to calculate the specific leaf area, you need the leaf area and the dry mass, and to calculate leaf dry matter content, you need your dry mass, the dry mass and the fresh mass. I also did an analysis called relative interaction intensity, or RII, which is going to help us figure out our plant interactions. So the calculation is really simple. You've got your paired individuals in a plot, the one with neighbors and the one without. You do this simple calculation, and then the output is either going to be a range of values from -1 to +1. So if it's -1 or close to -1, it's a competitive interaction. If it's 0 or close to 0, it's a neutral interaction, and close to +1 would be a facilitative interaction. And these are usually calculated with plant growth metrics and biomass, and I used plant height growth per day and leaf count growth per day for this calculation. To do those growth rate per day calculations, I took the plant's end height and beginning height, divided them, and then divided that by the number of days that it had lapsed, and the same for leaf count. In terms of my analyses, I used multiple statistical tests. So to analyze my survival data, I used a chi-square ANOVA of a generalized linear model and logistic regressions. And I used linear models to analyze my plant growth metrics, abiotic conditions, and leaf trait data. And then I used an ANOVA to analyze our plant interaction data. So jumping in, what did I find? We're going to start with the abiotic results. And this graph we're looking at is showing the average soil moisture in each pasture. So we've got our pastures on our x-axis, split up by our competition treatment, and our average soil moisture on our y. So what we're seeing here is pastures 3 and 4 had the lowest soil moisture. Pastures 1 and 2 had the highest soil moisture, and the soil moisture significantly differed from one another in pastures 1, 2, and 4. We also see here that plants without neighbors consistently had higher soil moisture than their counterparts with neighbors, although it was not significantly higher, but it was consistent at each pasture. Now we're looking at basically the same information, but instead with our PAR data. So the amount of sunlight reaching our plants. And so here we saw that individuals without neighbors had significantly greater PAR on average than those with neighbors. We also saw pasture 4 was significantly different than the others, most likely to this pasture due to this pasture having the tallest vegetation out of them all. I also want to point out that pasture 3 has the most intense or stressful conditions out of all the pastures. So here we see the highest PAR readings out of the 4 pastures and the lowest soil moisture. And we didn't know this before doing the study of which would be the most stressful, but we deduced it from these 2 graphs. Now we're looking at plant survival over our study period. So we've got our days elapsed on our x-axis and our percent survival on our y. So, and then each line is a different pasture, and the black line is all the pastures combined. So overall, we had about 77% survival. But the pastures significantly differed from each other, and we can kind of see that here. Like pasture 3 lost about half of its individuals by the end of the study, whereas like pasture 1 had no plant death. It's not clear why the pastures differed from each other or what factors influenced that. Here we're looking at how soil moisture influenced plant survival. So this is a logistic regression of plant survival and the average soil moisture. We can see that the probability of survival increases as soil moisture increases to 40%. And our survival here is not a percentage, but rather a binary. So 0, the plant did not live, and 1, the plant survived. There's a suggestion in the model output that the soil moisture influenced survival, but it wasn't significant. Here we're looking at soil moisture and how it influenced leaf growth. So we're seeing leaf growth per day on our y-axis and average soil moisture on our x. And we're seeing that as soil moisture increases, so does leaf growth in our plants, and that was significant. Now moving on to our competition results. So here we're looking at our competition treatment and our plant height growth per day. So we've got no neighbors and our neighbors present. And here we can see plants with neighbors grew significantly taller. And then when we break it down by pasture, we see it's true in all of them except for pasture 1. And the effect was really strong in pasture 3, where our abiotic conditions are most intense or stressful. Now we're looking at our competition treatment and our leaf dry matter content. So here we're seeing higher LDMC in our individuals with neighbors. It wasn't statistically significant, but it was suggestive. So plants with neighbors had higher LDMC. And we also saw that Dicanthelium had significantly higher LDMC than Axonopus, which makes sense. Dicanthelium has these smaller, thicker leaves, and Axonopus has kind of larger, broader leaves. Now we're looking at our plant interactions. So remember that RII is our calculation to look at whether our interactions are competitive or facilitative, and it's on the scale from -1 to +1. So that's our interaction strength we're seeing on the y-axis. And we've got it split by pasture and also by species. So the darker gray is Axonopus, and the lighter gray is Dicanthelium. And we can see here that our plant interactions are varied. There looks to be possible facilitation over here in pastures 2, 3, and 4, but it's a little varied and unclear. Pasture 3, which again was our most stressful pasture, had a really strong facilitative interaction for Dicanthelium. And again, our pastures significantly differed from one another. And oh, I forgot to mention, this is the plant interactions that were calculated using our plant height. So this graph got the same information, but it was calculated using our leaf growth metrics. So this one's even more unclear and kind of all over the board in terms of we have a mix of neutral, competitive, and facilitative interactions. So we didn't see consistent responses among either growth metric or among either species. And we think a better metric to understand this would be biomass, as it measures plant productivity, but it wasn't collected for this project as it's a destructive collection method, and I was interested in following plants throughout a study. So what does it all mean? So first I'm gonna go through our abiotic results. So first, there was really low evidence for an effective PAR on the plant growth metrics. This could be because that even with neighbors present, sunlight was just very available and not a limiting factor to our plants. Our soil moisture hypothesis also wasn't supported. We saw greater survivability when soil moisture was high, which was peak wet season. So in some of the pastures that looked like an inch or more of standing water, which I predicted would cause plant death or a decline in growth. So the data didn't follow our predicted nonlinear relationship. Soil moisture did affect plant survival and leaf count. So there's preliminary evidence that changes in soil moisture is going to impact these plant communities. But it is important to mention that we only looked at a small part of the soil moisture gradient, as all the data was collected in the wet season. So as a result, we only have part of the picture of how soil moisture affects plant growth. We also saw no variables influence either species specific leaf area, which is a little surprising. We didn't see changes in SLA along with LDMC, but it's congruent with the theory that LDMC is harder for plants to modify. Excuse me, LDMC is easier for plants to modify. It's a more plastic trait than SLA is. Also, the fact that I started with fully mature plants and it was a short-term study could also explain why we didn't see significant changes in SLA. Our hypothesis that we would see increased LDMC and reduced SLA when soil moisture was high was not supported by the data. We saw no significant effects of soil moisture on LDMC. It could be that plants here are evolved to adapt to dramatic changes in soil moisture and standing water. And so while that's a common plant response elsewhere, it's just not here. Could also be that because all the data was collected in the wet season, the change in LDMC might have already occurred from that shift from dry to wet season. So I missed it in my study period. We did see that the pastures had variable conditions. Some were more stressful than others, and there was evidence that it affected the plant interactions, but it was variable. And this is why the rainout pastures were intentionally placed on a gradient. We wanted them to be representative of a lot of different conditions. And plant interactions and how the different rainout treatments are going to alter them is going to be really important to follow over time, as they're going to give us insight into the resiliency of our plant communities to changes in climate. In terms of our competition treatment and how it influenced our plant responses and plant interactions, we thought we would see greater growth in neighborless plots, but we actually saw the opposite, with greater growth in plants with neighbors. And we're not exactly sure what caused this, but to understand the outcome of these interactions, we think biomass needs to be sampled in addition to measuring the other growth metrics. Our hypothesis that plants with neighbors would have higher LDMC and plants without neighbors would have lower LDMC was supported. So this could mean that if the rainout treatments cause more competitive plant interactions that then influence and change plant leaf traits, this could have larger effects on ecosystem functioning due to the additive effect of competition and resource limitation. One of our main consistent results was that the pastures differed from one another, and it was a very strong effect. What's happening in one pasture won't explain what's happening in another, and we could see highly localized community response in each of our pastures and treatment plots in the rainout experiment. So we'll see as the experiment continues, but that concludes my talk. Thank you all so much for listening. Before I take questions, I'd love to just extend some big thank yous. First to Betsy Boughton for her assistance and guidance throughout the entire process of this project. I always felt really supported by you, and I've appreciated your help so much. I also want to thank Lydia Landau for being an amazing mentor and friend to me. I feel so lucky to have gotten to work with you every day on the Rainout project. I also want to thank Greg Saunier for his endless patience when teaching me about leaf traits, and also for all your help with R. I want to thank Shukai and Hao Yu for their help with project design and instrumentation help. And I also want to thank the entire agroecology lab. I really felt supported by all of you during my time here, and I'm going to miss working with all of you so much. I also want to express my gratitude. Oh my gosh, I'm not going to cry on Zoom. I want to express my gratitude for my research being supported by a National Institute for Farming and Agriculture grant awarded to Dr. Chu and Dr. Boughton. Okay, I'm ready for questions if there are any.
Great job, Flynn. Thank you. There are some questions, and one is from Meg. She asks, what is the elevation difference between the pastures 1 and 4? Like, and I don't know if you remember that particular
I
detail.
know that off the top of my head, no, but it's probably not the most dramatic. I don't know.
No, I actually looked it up while you were talking, just so I could be prepared. It's about 73 centimeters, so it's not huge.
Yeah.
Here's a question from Lydia. Did you see any correlation between plant survival and whether or not it had neighbors? Do you think clipping around plants had any impact on survival?
That's a really good question. We did look at that and it didn't come out as a significant effect. I was kind of wondering about that during the study though, but we didn't see an effect of that.
And here's a question from Josh Daskin. He says, very well presented, Flynn. Did neighbor removal affect soil moisture? Could that have contributed to the neighbor facilitation?
Can you say that one more time? Sorry.
Did the neighbor removal affect soil moisture? And could that have contributed to the neighbor facilitation?
I believe it did. We go back. So our plants without neighbors had higher soil moisture, but it wasn't significant. So it might have, but it wasn't a strong effect. And could it have contributed to the neighbor facilitation? I'm not sure, but that's a great question.
Here's another question from Amy Lowe. Do you have any hypotheses as to why pasture 3 was so wonky compared to the other pastures you were looking at?
Not specifically any reasons. It was like in the middle of the elevation gradient. And I wouldn't say its vegetation was super different than the other pastures. So it's kind of unclear.
And last question from Mandy West. How much change have people seen in SLA for a specific plant?
I'm not sure how much change we would see in terms of like a metric to give you. I don't have that.
That's a good question.
Yeah.
All right, well, that's all the questions for now. Thank you, Flynn, and good luck in your next endeavor in Montana, and we'll miss you.
Gonna miss you all too. Okay, I'm gonna mute and stop my video to let Maya switch on.
Okay, thank you. All right, our next speaker is Maya Zambrano-Lee. Maya graduated from the University of Maryland with a Bachelor of Science in Environmental Science and Policy. And before coming to Archbold, she worked in environmental education in Alabama and also conducted a research experience for undergrads on the costs and climate-related benefits of phasing out hydrochlorofluorocarbon refrigerants, and also as an invasive species removal intern and a rafting guide. Maya has been a really fun addition to our lab, and we're going to miss her curiosity and fun questions for sure. She always had lots of good questions. Today, she will give a talk entitled Assessing the Long-Term Recovery of Plant Communities and Soil After Ecosystem Modification. And Maya is getting her presentation up and going.
Can you see me?
Yes.
How do I change my name from being Flynn?
I think
I'm gonna try to, yeah, I'm gonna see if I can help with that.
Yeah. All right, just 1 second. I'm gonna share my screen.
Did I spell it right?
Yes,
Okay, awesome.
thank you.
Yep.
All right, thanks, Maya. Over to you.
Thank you. Can you see my screen?
Yes, I can see it.
Wonderful. Hi, everyone. Thank you so much for coming to my presentation. Thank you, Betsy, for the introduction. And today I'll be discussing two types of ecosystem modification, which are nutrient addition and wild hog rooting. So grass-dominated ecosystems account for up to 40% of all land on Earth and are critical habitats for biodiversity, water quality management, carbon sequestration, and agricultural production. And grasslands undergo many forms of modification, and one that is of international interest is nutrient deposition and just the modification of the nutrient cycle. So additional nutrients may be introduced into a system via atmospheric deposition, rain, smoke, or fertilization. And modifying nutrient availability can have effects on soil and vegetation in natural and working landscapes. So here in Florida, another driver of grassland modification is feral hogs. Wild hogs are an invasive species in many parts of the world, and they can cause a great deal of damage, digging up plants and uncovering the bare soil and altering plant communities. And in the US, they cost over $1 billion every year in damages and control. So as total grasslands worldwide has decreased over the past century, there's a big need to understand how to best manage remaining grasslands against potential threats to healthy ecosystem functioning. And here at Buck Island Ranch, a formal experimental site, which is the large star down there, called the fire plots, were modified by nutrient deposition and hog disturbance. So my intern project focused on revisiting that site several years after the modification events occurred to study potential long-term impacts. So first I'll give a brief history on the fire plot experiment, which was started by Dr. Bolin in 2002 and taken over by Dr. Boughton in 2010. And then I'll take you through my first study on whether nutrients added as fertilizer have persisted within the soils of the fire plots. And then I'll go over my second study on how plant communities in those plots have changed since a feral hog rooting event. And I'll summarize my results. So we'll start with the fire plot experiment, which ran from 2002 until 2016 and was on seasonal fire and nutrient addition, specifically looking at how do plant communities and their species composition respond to season of fire or prescribed fires in the summer or winter, and phosphorus and nitrogen fertilization. So the site that was used for the fire plot study, and then also my two studies, is a semi-native subtropical wet prairie that was released from cattle grazing in 2002 and then was fenced off to exclude further animals. And it had never been fertilized or plowed. The vegetation communities are dominated by perennial native grasses, and the soils are sandy, acidic, and poorly drained. So this map on the right here shows the setup of the fire plot experiment, which remains in place today, and it's a split plot design that has 12 plots. This is a plot, and they're arranged randomly in 4 rows containing 3 types of burn treatment. So those letters right in the middle, that WB stands for winter burn, meaning the prescribed burns were in the winter. SB for summer burn, and then CB stands for control burn, or that plot remained unburned. And then within each plot, there are 4 subplots and 4 nutrient treatments. And each subplot either received nitrogen fertilization as N, nitrogen and phosphorus fertilization as NP, phosphorus alone as P, or no fertilization listed here as C for control. And these treatments went on almost every year from 2003 until 2016. So looking at the timeline of how this all is together, in 2002, the plots are released from grazing and the experiment began and ran relatively smoothly until 2012, when hogs breached the fence and rooted up a bunch of the plots, and then a native plant called red root broke out everywhere where the hog rooting had happened. And that disruption ended the seasonality of fire study, and then the fence was repaired, and treatment and data collection still continued until 2016, and the plots were more or less left alone. Then a few years later, in 2018 and 2020, or 2018, 2019, and 2020, the entire pastures were pasture was burned, including the control plots, as part of nearby prescribed burns. And then in 2022, I conducted my intern project on ecosystem modification. So the first modification we're going to look at today is the intentional one caused by the fire plot experiment, which will be the 13 years of nutrient addition and looking at whether added nutrients persisted in the soil. So my research question was, what impact does 13 years of nitrogen and phosphorus fertilization have on soil nutrient levels after 6 years without fertilization? And as I mentioned, there's a need for this research to understand how nutrients might persist on grassland soils after fertilization ceases. So I hypothesized that for 2022 soils, nitrogen would not significantly differ from any of the plots, and because we know that plants around here are generally nitrogen limited and are quickly to use available nitrogen. However, I hypothesized that in the subplots that received phosphorus fertilization, they would have higher levels of plant available phosphorus compared to those that didn't receive phosphorus fertilization. Because Central Florida has a history of high levels of phosphorus in agricultural soils ever since excessive fertilization in the 1900s. And plants don't need very much of it, so added phosphorus is more likely to persist in soil. And lastly, I predicted that soil organic matter would be higher in the nitrogen fertilized subplots because some research has been found that nitrogen increases biomass. So I took 12 samples total of the soil and 3 replicates for each nutrient treatment from the highlighted plots here, and we went about 15 centimeters deep. And the map in the middle shows where I sampled, and it relates to this map on the right, which is a map of where the hogs rooted, and that's shown in black and gray. And so we were avoiding the rooted areas from where they'd been rooted in the past, as well as avoiding places that had currently been rooted, because it seems that the hogs have since broken back into the plots. In the lab, I tested for plant available nitrogen as ammonium and nitrate, plant available phosphorus as orthophosphate, and soil organic matter, or SOM. And then we compared the 2022 data with the fire plots studies data from the years when soil was collected in our studio, and conducted normality tests on our data and normalized some of them. And then for the normal data, we ran repeated measures, split plot ANOVA tests with the response variable being a function of year plus the nutrient treatment plus year interacting with nutrient treatment plus error as the block or whatever row the plot was in. And we didn't find any year interacting with nutrient treatments throughout the whole study, so we'll just discuss year and nutrient treatment as effects by themselves. And I also ran multiple pairwise tests to better understand the differences between treatments and years. And then for non-normal data, we ran Cresswell-Wallace tests. So here's what we found. First, as I go through my results, please remember that all of these are preliminary due to their small sample size, because I only had 3 replicates for each treatment. So as we look at the next couple graphs, note the y-axis is, the x-axis is year that the soil was collected, and the y-axis is ammonium, or micrograms of ammonium per gram of soil. And this bar graph shows the average amount of ammonium in each type of treated subplot on a given year. So the colors are associated with the nutrient treatment those subplots received. So in this graph, for example, when we look at the yellow bar, we're seeing the average ammonium levels among the 3 fertilized phosphorus fertilized subplots, but not in this one, we're not looking at phosphorus. So just to clarify. So for ammonium, we found that there was a significant year effect, specifically that there was significantly less ammonium in 2002 than there was in 2004 and 2006. However, we found no effect of fertilization, meaning that there was no significant differences in ammonium levels, regardless of what nutrient addition treatment the subplot received. And so then when we look at nitrate, we see a similar trend in that there was a year effect, but it's highly variable, meaning that some years were like higher or lower than the previous year. And so there's not a clear trend. although 2022 did have significantly less nitrate in all the plots because the soils came back with no nitrate detected. And again, we did not see any effect of fertilization. So these results support my hypothesis about nitrogen fertilization not persisting in the soil. And I believe the reason we don't see a nutrient effect in nitrate or ammonium is because plants used the fertilizer quickly after it entered the plot, so none would have been left over to persist in the soil. And in general, wet prairie soils in Florida have low nutrient contents. Additionally, the decrease in 2022 soil, 22 nitrogen levels compared to 2004, especially in ammonium, could be due to the removal of cattle as an input of nitrogen. Cattle are fed supplemental feed, and as they grazed these fire plots, they were contributing manure and urea to the plots prior to 2002, and it's possible that that may have had an effect by adding that nitrogen earlier. And then lastly, lower 2022 nitrogen levels may be a long-term effect of the annual burns that happened, as it's well known that burning vegetation is a major way that nitrogen is lost from these ecosystems to the atmosphere. All right, so moving on to phosphorus, here we're looking at micrograms of orthophosphate per gram of soil. And we did find the year effect to be significant. So from 2004 to 2006, only the phosphorus subplot increased, the phosphorus only fertilized subplot. In 2006 from 2011, again, only the phosphorus subplot increased. But if you look directly from 2004 to 2011, there was actually a significant increase in the control, the phosphorus and nitrogen and phosphorus subplots. So then fertilization stopped in 2016, and when we compare 2011 to 2022, we see significant decreases in the nitrogen and phosphorus subplot and the phosphorus only subplot, with a small increase in control. So we also saw an effect of fertilization, showing that on average, the phosphorus and the nitrogen phosphorus plots had higher levels of orthophosphate than the other two plots that didn't receive phosphorus fertilization, suggesting that the added phosphorus was indeed building up over time. And then when they stopped fertilizing in 2016, we can see that there was a decrease from 2011, but not completely, as the levels of orthophosphate in 2022 are actually not significantly different from 2006 at all. And so what that tells us is that fertilizing with phosphorus did have a long-term impact, because 2006 had higher phosphorus levels than 2004 had had. So some of the reasons we think this may be is that these soils may have a higher storage capacity of phosphorus. This wet prairie, the study site, is in a lower lying area of the ranch that has like a slightly muckier top level of soil with more aluminum and iron that allows phosphorus to better bind in that top layer of soil. However, that only explains why we see any remaining phosphorus today. And so where did the rest of the orthophosphate go since 2011? Definitely some of it, or a notable amount, would have been used by plants, especially in the nitrogen and phosphorus plots, because plants that aren't limited by nitrogen are able to take up more phosphorus. And then when the fires came through and burned plant matter, regrowth likely demanded additional uptake of phosphorus, because some of it may have been lost to the atmosphere. And then some of the phosphorus likely leached or ran off. Beneath the O horizon are pretty sandy soils that struggle to bind to phosphorus, and studies in Florida show that seasonal flooding in sandy soils have like changing heights of the water table, and that can contribute to loss of phosphorus to leaching. So now for soil organic matter, note the change in the y-axis is soil organic matter is a percent of all of total soil. And so we did find a significant year effect. So 2004 and 2006 were not different from each other, and 2011 and 2022 were both significantly lower than, or had significantly lower soil organic matter than 2004 and 2006, except for the phosphorus subplot in 2022. And the only significant change since fertilization stopped actually was that the control plot increased in soil organic matter. So there was also a significant fertilizer effect as well that suggested that nitrogen-only subplots on average had less soil organic matter than phosphorus-only subplots, which was contrary to my hypothesis. So there was a lot of variability in soil organic matter overall, which makes it difficult to say conclusively what caused these initial changes. But my research has led me to believe that the initial years of higher soil organic matter may have had to do with grazing. Grazed plants invest more into their root structures. And so as time went on without grazing, there may have just been less plant investment into their roots, explaining the decrease we see here. And another potential explanation lies with fertilization treatment. Research has shown fertilization can increase or decrease soil organic matter depending on the environment. And some studies I found showed that nitrogen and phosphorus fertilization, but especially nitrogen, can initially increase soil organic matter, but over time leads to increased microbial activity as microbes have like higher food supply, leading to what some might call a microbial feast. And then soil organic matter goes down. And the last potential theory is that plant species over this time were changing due to the release from grazing and the fire plot treatments. So different species might yield different levels of organic matter. So that concludes study number 1. Now we're going to move on to study number 2, get ready, plant stuff. So back at our timeline, now that we've analyzed the intentional nutrient modification, we're going to investigate the unintentional modification that occurred in 2012 in basic feral hog brooding. So we have species data from up to 2015. So we're going to look at how the hogs messed up these plots. In 2012, when the hogs broke through the fence and rooted the plots, you can see that where they rooted on this map on the left in the areas in black and gray. And then in response to the rooting, a native forb called Lachnanthes caroliniana, which I'll also refer to as Carolina redroot or just redroot, colonized over 90% of the rooted areas. And so everywhere on black on this map is where redroot grew after the rooting event. And these 5 plots had the most rooting per burn treatment. So those are the ones I focused my study on. And redroot reproduces clonally and loves disturbance. And in 2012, it became nearly a monoculture in these plots, which had completely shifted away from the grass-dominated communities that were there before. So I wanted to find out if this community change to redroot is permanent, which brings us to my second study on plant community recovery. So now that it's been 10 years since the redroot outbreak, I was curious, how have plant communities bounced back from a monoculture created by hog rooting? Did this one-time hog modification like permanently change the communities in the plots, creating an alternative stable state, or did the plots return to their previous compositions? So there's a need for this research because most studies on hog rooting don't return long after rooting events, and even fewer have pre-rooting data. So the study is unique in that it provides insight into long-term recovery. And it's also helpful to know how impactful a single rooting event can be on plant communities, to know how urgent like the need for hog management might be. Here at the ranch, there is some hog selling and hunting, but we still have a substantial hog population. So my hypotheses were that Carolina redroot would no longer be the dominant species due to the fence excluding further hog damage, and that plant communities would return to their pre-rooting composition. So like I mentioned, I focused on these 5 highlighted plots that had the most rooting, 3 of which were control burned, so they hadn't been burned since before the 2002 and until 2018. We sampled 10% of each subplot, estimating the percent canopy cover of each species we observed using a modified Daubenmeier scale on the right here. And during our analysis, we were testing for changes in dominant species and their abundances over time, as well as shifts in overall composition of the plant communities over time. And then in our studio, we conducted a non-metric multidimensional scaling ordination to compare community composition and ran repeated measures split plot ANOVAs, looking at dominant species cover as a function of year plus nutrient treatment plus year interacting with nutrient treatment plus error as the block that they were in. And so first, when we look at this graph, note the year is, x-axis is year and the y-axis is percent cover of a given plant, showing us how much of the subplots on average did that species cover. And what we found supported my initial hypothesis that after the hogs rooted in 2012, red root has significantly decreased. You can see that in the following 3 years after the rooting event saw a steady decline. And in 2022, it's at the lowest point it's been in 15 years. So if red roots decreased, what replaced it? We noted as well that there was a shift in the dominant grass species over time, that they've been changing, with panic grass or Panicum longifolium on the left having generally been declining since the beginning of the experiment. And then species of blue stem on the right have also been varying quite a bit. And then when the hogs come in, you can note that it seems blue stem was able to like seize the opportunity to become the dominant grass in over that time. And it's now averaging 45% coverage in the plots we sampled, whereas panic grass has stayed below 20% coverage on average. And then another change we saw in non-dominant species was a significant increase in 2 forbs, on the left, pine barren goldenrod, and on the right, Hediota siniflora, and a species of sedge within the Rhencosper genus. And you can see they've all been going up since, or they're all higher than they were in 2015. And so as we remember that 60% of the plots that I sampled for species composition were unburned from 2002 and 2015. And so I believe that this effect doesn't actually have as much to do with the hogs, if you like look closely at where 2012 would be, as it does with the fires that happened in 2018, 2019, and 2020. As we know that fires are critical for plant biodiversity in Florida. And so lastly, I'd like to go over how plant communities as a whole have changed over time. So now when you look at this graph, please ignore the axes you see right now. I'm going to give you new ones to focus on in a minute. This is a species ordination graph, and what we're looking at is the spatial relationship between the years. Each year has all these points coming out of it, and each point represents a surveyed subplot for that year. And then the years shown here basically represent like the average, like an average subplot for that year in terms of like what the species compositions were. So like an average composition of the subplots for that year. Now, the closer 2 years are to each other, the more similar they are. And then the further apart they are, the less similar. And then movement up and down, left and right on the graph is correlated with the presence of different species and abundances. Movement to the right of the graph is significantly correlated with higher amounts of red root, and movement to the left of the graph corresponds with higher amounts of panic grass. Now, if we look back at 2002, when the plots were released from grazing, we see that once like the fire treatments begun, the plot compositions showed a big change to 2003, and then another big change to 2004, and they really didn't like kind of become steady or like stable until 2009 through 2011, where the plots seemed more stable, like and resembling each other 3 years in a row. Then we have 2012 hogs break into the plots, and this shift to the right, which correlates with that higher red root in those plots. And so we see none of the other plots are as far on the right as 2012. That's the year that had the highest red root. And then when we look to 2013, there's a shift back to the left, and then the years are changing a bit. But this shift back to the left does indicate like a partial recovery from the rooting event, and like partial return to pre-rooting conditions. And then we also see that 2022 is significantly different from all previous years. We know 2022 had higher levels of blue stem grass and forbs in the sedge that I mentioned. So we can know that part of the movement down the graph here is correlated with higher amounts of those plants. So the first reason I believe we see partial recovery in the immediate years after the rooting is due to the fence excluding further hog damage. And these ecosystems are like bounce back from recovery in general pretty quickly. And so that the hog rooting exclusion may have given them enough space that they needed to go back to their previous composition. And some of the reasons we think that 2022 changed so dramatically is that since 2016, the plots have stopped receiving fertilizer. They were burned 3 times. And the composition of these species have just been like shifting along with all those, along with all of those changes. And then another factor that potentially explains continuous movement all over the graph is the fact that these ecosystems have been adapting to the loss of grazing. And potentially there's been a shift to plants that better thrive in the absence of frequent herbivory. And so lastly, I want to mention that the hogs have broken back into the plots, and I saw red root growing on top of recently rooted soil. So perhaps these plant communities can partially recover from a single hog rooting event, but further and more frequent modification could threaten composition moving forward. So to conclude, summarize my results. In my study on modification by fertilizer and soil recovery, nitrogen fertilization had no effect and did not persist in the soil. Phosphorus fertilization did seem to persist in the soil, and more research is needed on soil organic matter to understand how it's impacted by the combination of grazing, fire, and nutrients. And then in my study on modification by invasive hogs and plant community recovery, we found that plant communities did seem to recover from the red root monoculture, but they did create somewhat of an alternative stable state with a shift in the dominant grass species. But it's possible that that may have happened anyway, since the panic grass had been decreasing. And communities changing. Plant communities change from long-term grazing removal and fire. So putting these results in the context of grassland management and recovery from modification, we see that phosphorus persisting long-term in the soil means we need research on how to restore soils affected by nutrient persistence, especially given that excess phosphorus has a tendency to degrade water quality. And additionally, my results showed that grazing can almost be like a form of ecosystem modification, not in any like bad way, but just that as we removed grazing, it seems that it also altered plant communities long-term and potentially the soils as well. And lastly, we know that wild hogs can create major disturbances, and at least partial vegetation recovery is possible with hog management, and fire may play an important role in supporting that recovery. And so that concludes this part of my presentation. And so before I move on to questions, I would like to thank, oh my goodness. I would like to give a big thanks to Dr. Betsy Boughton and Gregory Saunier for all of their help in my R and just my research, my data collection, and everyone on this list. Truly, you've helped me so much with my project and also just made my time here at Buck Island Ranch incredible. I almost started crying during Flynn's presentation, so I'm not going to do it now, but thank you all so much. And thank you to the LTAR grant for funding my stay here. And here's my references. Does anyone have any questions?
Good job, Maya, you did it. So complicated project with two sub-projects. So, great job.
Hope everyone was able to keep up. I know there was a lot.
There are some questions. So Lillian asked, Lillian here from Maryland, what was the purpose of the burn of all the plots, including the control plot from 2018 to 2020?
I believe that it was just, Betsy, if you could actually help me with that one, was it just they were doing prescribed burns anyway, and the fire plot study was not really being like actively pursued?
Yes.
Yeah.
So it was just part of a prescribed fire that was in the pasture around the plots, and we didn't have any reason to keep the season of fire treatment going at that point, so we just let it burn through all the plots.
Yeah, there's prescribed fires going on in the pastures around here like all the time in the winter.
And Lydia asks, why do you think there was less red root in 2002 through 2007 than there was in 2009 to 2010? Do you think the red root population would have become as dominant if the rooting event had occurred in 2006, for example, when the red root population was lower as compared to 2012?
That is an excellent question. I do know that there was like some drought conditions that were like, or some drier soils that may have had an impact on, because like there was some change in vegetation during that time. I think it was like 2007, 2008-ish. So it's perhaps red root liked those conditions, and that's why it had started increasing before. But that's a guess. I'm not entirely confident. Now, whether or not, I'm not sure if there would have been as heavy of a like monoculture outbreak if it had started earlier, because there simply wasn't as much of the red root already present. It is interesting though that hogs seem to be drawn to red root, so it's possible that red root was growing, which is why the hogs came, because they like to eat it, and then it grows in response. So it's kind of like a positive feedback loop. So if there hadn't been as much, maybe it wouldn't have drawn the hogs in as much either, if that makes sense.
Yeah, I think that is a great explanation, and maybe it was like the slowly increasing red root over time, and then that attracted the hogs, and then they rooted it. So yeah, interesting. And there's a comment from Betsy Rothermel. Very interesting, Maya. You did a great job explaining and interpreting your results.
Thank you. I tried super hard.
Yes. Mandy says, what's your favorite plant?
I've lost sight of every plant in my favorite bird is sandhill cranes. It's not a plant. I really like slash pines. Large trees. I'm so sorry. I'll get back to you on that.
Great. Thank you everyone for coming, and thanks Maya, and good luck in your future endeavors.
Thank you. Thank you everyone for listening to me and Flynn for like an hour. We appreciate it.
Great job, both of you. So thanks everyone who joined us today. Next month, join us again on April 28th for another agroecology seminar presented by Akeem Go. Bye everyone.
Bye.